Okay, so good afternoon, and thank you for I know it's late, so I'm going to try to be brief, and it's always hard to talk after Dan because it's a really tough act to follow, but the good thing is that he actually explained a lot of the concepts in tumor evolution that I'll be touching upon, so I can, allows me to be more brief. So I have no disclosures, and I'm a medical oncologist, a genitourinary medical oncologist. I spent most of my time in the laboratory studying urothelial carcinoma, and the reason I study urothelial carcinoma is that it's a big clinical problem, which, as you can see from this graph, is the cancer-specific mortality for urothelial carcinoma. And this is in many other cancers is supposed to be a curve, whereas as you can clearly see here, it is a flat line because we really haven't changed the outcomes from urothelial carcinoma over the past 3 decades, and I think we all agree that our patients deserve better. So that's why we started studying this disease, and for the past 30 years, chemotherapy was the mainstay of treatment for advanced urothelial carcinoma. And on this slide, you can look at the overall survival. And as you can see, we have really been unable to improve the survival from using these different combinations of chemotherapy. In the second-line setting, we really had no effective therapies for the past 30 years until the first immune checkpoint inhibitor was approved last year, which is obviously something that we're very excited about. However, it is important to keep in mind that actually less than 30% of patients, actually close to 20% of patients, respond to immune checkpoint blockade. And obviously, we need to do better, but as Dan just mentioned, the issue is that many patients who have initial responses to chemotherapy or even immunotherapy will subsequently develop resistance. So in this context, we decided to investigate resistance to therapy from an evolutionary standpoint, and we designed our study to answer 3 specific questions. The first is, what is the degree of clonal divergence between primary untreated and advanced chemotherapy urothelial carcinoma? Second, how does the chemotherapy really shape the genomic landscape of urothelial carcinoma? And finally, what are the mutagenic mechanisms that drive this evolution? So this is our paper, which we published earlier this year, and basically what we've done is we prospectively collected 72 urothelial carcinomas from 32 patients, and our cohort included trios of matched primary, metastatic, and germline samples, and we collected pre-chemotherapy primaries and matching post-chemotherapy tumors from all the metastatic sites for urothelial carcinoma. We also obtained multiple tumor samples from 2 rapid autopsies, which is a way for us to really study how the tumor evolve over time because we actually have to go in and get the tumor. We don't have the luxury of sampling the blood to get that deep kind of understanding yet, although it's something that we're working on. And most patients in our cohort were smokers, as expected for patients with urothelial carcinoma. Most were males, and the majority developed metastatic disease during the study period. And we performed whole-exome sequencing of the tumor and germline DNA, and as was briefly touched upon earlier, we really needed an algorithm to be able to adjust for the tumor purity and clonality over time to be able to compare the allelic frequencies of different mutations over time. And this is work that was done by our collaborator, Francesca De Michaelis in Italy. So the first question, what is the degree of clonal divergence between treatment-naive and chemotherapy-resistant urothelial carcinoma? And I think you may have seen this earlier. And as you can see, there is really a striking degree of heterogeneity, and that was really preserved between primary tumor samples and primary metastatic tumor samples after chemotherapy, showing that it's probably really related to the effect of chemotherapy rather than a feature of the tumor biology itself, although there are probably elements of both. And surprisingly, even mutations in well-known driver genes were not always shared between the pre-chemotherapy and the post-chemotherapy tumors. So sometimes we even detected two different mutations in the same driver gene. So for instance, in this patient here, we identified the pre-chemotherapy tumor sample had a particular p53 mutation. At the same time, the post-chemotherapy lung metastatic lesion had a private mutation in the same gene, but a different mutation. And interestingly, both of these mutations were subclonal. Actually, I think these moved a little bit. I'm talking about the p53. I'm sorry. So both of them were clonal. This one and this one. So in contrast, the copy number alterations appeared to be acquired early and actually to a large degree appeared to be clonally static during the lifetime of the tumor. And we observed this interesting clustering, which we validated in the larger TCGA database. We're working more in understanding what do these events mean in the evolution of the tumor. And to understand the sequence of events in each patient, we, within the lifetime of the tumor, we reconstructed the phylogenetic trees from each patient, and we calculated the evolutionary distance between the primary tumors, which are represented here as gray circles, and the metastatic tumors, which are represented as the solid black circles. And surprisingly, when we did this, we found that in virtually every reconstructed tree, the primary tumor was positioned as a branch. And this indicates to us that there is an ancestral clone that gave rise to multiple cell populations that continue to evolve in parallel, and that more importantly, from a clinical standpoint, that this branching evolution occurred very early and seemed to be rather the rule rather than the exception in these patients. And again, this is unfortunate for my patients, and I think about that every day when I see the patients in clinic. So because this is supposed to be a case presentation, I will tell you a little bit about one patient. And we focused on that patient to sort of understand the full evolutionary arc of this patient's cancer. And we took a longitudinal genomics approach to follow the patient from the time of the diagnosis until death. So this patient was initially diagnosed with muscle-invasive bladder cancer, received 4 cycles of standard of care gemcitabine cisplatin prior to a radical cystectomy and a lymph node dissection, which is also standard of care. Unfortunately, he rapidly progressed, developed metastatic disease, was started on a clinical trial with docetaxel and ramucirumab, and eventually passed away and had a rapid autopsy. And we obtained multiple tumor samples from this patient. So we had the primary pretreatment tumor from the TURBT, which is highlighted here in yellow. We had 4 areas from the residual post-chemotherapy tumor and a regional lymph node met at the time of the cystectomy. And then we also had 6 distant metastases that are highlighted here in dark green. So in total, we had 12 tumor samples and a germline blood sample from that patient that we collected, and all underwent whole-exome sequencing. And these are the results of our analysis. In this figure, each row here represents one tumor sample from a given site within this patient, and each vertical bar represents a given mutation in one of the genes that are listed on top. And the different shades of green represent the allelic frequencies, or the cancer cell fraction. And these are, again, corrected for tumor ploidy and purity, allowing us to compare across different tumors with different purity and ploidy. And we clearly, as you can see, we clearly observed significant heterogeneity in this patient, and mostly between the primary and the metastatic samples. And this heterogeneity really took one of 3 patterns. So first, we see these subclonal mutations in the primary that later become enriched in the metastatic lesions. On the other end of the spectrum, you see these really private mutations that are really unique to the metastatic samples, suggesting that these continue to evolve after treatment. And if you look here closely, you can see that, in fact, each metastatic lesion harbors its own private set of mutations. And finally, we identified these unique tumors that are unique to subclonal mutations that are unique to the primary tumors, but then disappear from the genetic pool of the metastatic lesions. So this was really eye-opening to us, and from this analysis, and again, we spent a lot of time on this trying to reconstruct exactly what happened on the molecular level to this patient, and basically this is our interpretation, and there are other interpretations, but if you look here, we think that the initial founder clone harbored early truncal mutations that I'm pointing to here, and they were all shared by all the descendant clones. And this is, again, a principle that Dan has just explained. And one of the earliest cancer cell clades, as you can see, had already separated at this divergence point and resulted in a population that metastasized to the pelvic lymph node. And then this lymph node was later dissected during the time of the cystectomy, and the surgical removal of that lymph node prevented those mutations from entering the genetic pool of the rest of the primary tumor and the metastatic lesions. Then branching evolution continues, as you can see here, and additional driver mutations were acquired, as you can see here, important ones such as P53 and TSC1. And interestingly, the TURBT bladder tumor had actually the furthest genetic distance from the other tumors, and this really means that in this patient's case, at least 5 waves of clonal divergence had already occurred at the time of the very initial TURBT that happened at the time of diagnosis. So again, that is very sad for the patients, but it was an interesting phenomenon to see. And many of these mutations were actually implicated in the cellular response to cisplatin, such as POLD2 and FOXP1. And after neoadjuvant chemotherapy, you can see that a lot of these mutations have actually disappeared from the evolutionary record as they were eradicated by neoadjuvant chemotherapy. So by the time the other important thing to note here is that, but probably by the time the cystectomy took place, distant metastatic spread had already occurred, and this likely originated from this tumor population that gave rise to this primary bladder tumor here and all the metastatic lesions. And then we could observe that this transition from the primary to the metastatic state was marked by an acquisition of a mutation in a gene called tetraspanin 8, which is a well-known pro-metastatic and angiogenesis-promoting gene. At the copy number level, our computational analysis predicted a subclonal heterozygous deletion of the tumor suppressor gene CDKN2A in the primary cystectomy tumor, and as you can see here, and this really evolved into a clonal homozygous deletion, i.e. loss of both copies in the post-docetaxel ramucirumab distant metastases, and as you can see here, we confirmed these results by FISH, again showing us that there's tremendous heterogeneity and evolution even at the copy number level. So on to our second question, which is really how does chemotherapy influence the genomic landscape of the tumor cell populations in advanced and metastatic urothelial carcinoma? And when we compared the density of non-silent mutations between the pre-platinum or the pre-chemotherapy and post-chemotherapy tumor samples, we found a significant increase in clonality of mutations in the post-chemotherapy samples. And this really suggests that platinum-based chemotherapy acts as an evolutionary bottleneck that restricts the mutational landscape of the tumor. And this is actually really interesting because our findings are really consistent with mathematical models. This is one that was published by the Vogelstein group back in 2015. And you can see here that basically the effects of chemotherapy on tumor clonality, where here you can see the treatment being applied. And as you saw earlier, there was many colors, each of which designating a clone. And as the tumor comes back, you see a significant increase in the clonality because what comes back are actually fewer clones. And that was consistent with what we observed. And we found that these clonal mutations in the post-chemotherapy urothelial carcinoma are actually clustered in specific pathways, more specifically the L1 CAM and the integrin signaling pathways. And this sort of goes back to a phenomenon that has been described before actually in hematological malignancies such as myeloma, which is called cell adhesion mediated drug resistance. And in this mechanism, integrins mediate survival and drug-resistant signals from interactions with the extracellular matrix, and this is a mechanism that potentially ties drug resistance to metastatic spread and the tumor microenvironment, and this is actually something that we're working on right now, investigating therapeutic targeting of integrin signaling by inhibiting a kinase called a focal adhesion kinase, which is downstream of integrins and a master regulator of this pathway of downstream signaling, and we've developed cell line-derived and patient-derived bladder cancer organoids. Lamont Barlow, who's a urologic oncology fellow in the lab, is actually working on this, and as you can see here, we clearly see organoid regression. We currently also have xenograft experiments that are ongoing that strongly supports the efficacy of this approach. So finally, we asked the question, what are the mutagenic mechanisms that drive the evolution of urothelial carcinoma? And as you can see, as you know, also cisplatin is the backbone of chemotherapy used for urothelial carcinoma. And previously, it was found to induce a specific pattern of C2A mutations in a C. elegans model that was very tightly experimental setting, controlled experimental settings. And we identified a very similar increase in C2A mutations after cisplatin treatment. And we then turned our attention to the ApoBec3 family of proteins, which is something that we're currently very interested in. And this is a family of 7 cytidine deaminases that deaminate cytidines to uridines. And each of these 7 enzymes have a specific preferred motif, and by looking at this particular motif, we were able to identify an increase in both the number and the clonality of ApoBec-induced mutations in chemotherapy-treated tumors. And our model for this is that cisplatin-induced double-stranded DNA breaks, and then which then results in having single-stranded DNA overhangs. We know that ApoBecs bind to single-stranded DNA. That's what to induce deamination, and then you get these C to T or C to G mutations. And again, just stochastically, some of these mutations will end up being mutations that confer survival advantage, and then that chemotherapy will lead to a clonal selection of these mutations, and that might explain why we see that these mutations are clonally enriched in post-chemotherapy tumors. So in conclusions, and this is titled Conclusions and Clinical Implications, I'd be happy to discuss both. Chemotherapy-resistant urothelial carcinoma is characterized by intrapatient and interpatient mutational genomic heterogeneity. The majority of the mutations are not shared, and that goes back to the idea that we need to do serial biopsies, or we need to develop technologies like circulating free DNA that capture the entire arc of a tumor's evolution. Branching evolution and metastatic spread are very early events in the natural history of urothelial carcinoma, and that confirms what we've known as clinicians for decades about the advantage or the survival advantage of neoadjuvant chemotherapy in bladder cancer. We do know that we have shown that chemotherapy-treated urothelial carcinomas are enriched in mutations in L1-chem and integrin signaling pathways, and that may represent a new therapeutic opportunity. And as I mentioned, ApoBec-induced mutagenesis is clonally enriched in chemotherapy-treated urothelial carcinoma, and we are studying this in much more detail to dissect mechanisms to potentially prevent or reverse resistance to chemotherapy. And finally, this is obviously a team effort, and this is the team at the Englander Institute for Precision Medicine, and thanks to the funding agencies and most importantly to the patients. Thank you.